I'm going to talk to you about how I interpret the Eolia trial, as Luigi mentioned. So rather than trials in general, try and focus on one. Clearly a very major publication, or in fact two major publications from the last year. And I'm sure you are all familiar with the Eolia trial, but I'll start by giving a brief overview of it. But I think the reason why this has been selected for a particular focus is that both the original trial and the subsequent reanalysis of it used some quite complex statistical methods. They looked at quite, the results are quite tantalizing, and there's been a lot of discussion and potentially criticism around the approaches. And hopefully, what I'll demonstrate today that in this case it's not the statistics that are at fault, and we can delve a little bit more into maybe why we don't see a significant result and what this might mean for any potential future trials of ECMO in severe ARDS or not. But starting off with a brief overview reminder of the Eolia trial. So the population was patients with very severe ARDS, so a PF ratio less than 50 millimeters of mercury for 3 hours, or less than 80 millimeters of mercury for 6 hours, or a pH less than 7.25 with a PCO2 of at least 60 millimeters of mercury for 6 hours, and they had to have had no more than 7 days of mechanical ventilation. The intervention was immediate. ECMO had to be initiated within 2 hours. Patients could be recruited from non-ECMO centres, but only if a retrieval team was available to be able to commence that ECMO within 2 hours. And the comparator was conventional ventilation, but with ECMO available as a rescue therapy within that group. And the primary outcome they were looking at was mortality at 60 days. So the design, where it starts to get a little bit more complicated, it was a group sequential trial design. So what does this mean? It means that they evaluate the outcome after a number of patients have been recruited and followed up. They used groups of 60 patients for this, and then repeat. So after every 60 patients, reanalyze the outcome and make a decision on whether to carry on with the trial or whether to stop the trial. We need to bear in mind here that group sequential trials are not actually a novel thing. In fact, most of our trials are really group sequential trials because any standard parallel groups trial which has pre-planned interim analyses is a group sequential design. We're used to seeing ones that have something like an O'Brien-Fleming or a Peto-Habital stopping rule. The difference here is that those rules are very strict. They will very rarely stop a trial early, and they'll only stop a trial when there is very strong evidence either that a treatment is effective or that it's harmful. The difference here is that Eolia used a more complicated and potentially more efficient design for group sequential trials. And I'll take you through what that design was and how it works. So, every time they analyse the data, they calculate two things. The first of these is a score, essentially called the efficient score. A value of 0 would mean there's no difference in the treatment. A positive value means that the new treatment is more effective. Negative, the conventional, the control is more effective. And then they would plot this against the amount of information. As you go through the trial, more information accrues, and we use a measure called the Fisher information for this. So each time there's an analysis, you can plot this score against the information. And then we can make some decisions from that on whether we stop the trial or continue. And for that, we use a set of stopping boundaries. The particular design used here was what's called the triangular design, or Woodhead's triangular design, because of these triangular stopping boundaries. And the first thing you can see from these is that unlike those traditional O'Brien-Fleming or Peto-Habital stopping boundaries, they are definitely not symmetrical. You have the ability to stop a trial even when it's showing some benefit, if it's not showing enough benefit. So at this top line here, if any of the results cross the top line, then we're showing significant benefit of the intervention, and we stop the trial at that point, declaring that it is effective. If we cross the bottom line, and are also below the score of 0, then we've shown that the intervention is harmful, and we would want to stop the trial. And then the interesting area we have over here is futility. So if we cross the lower stopping boundary, But at a point where the Z-score is positive, then we declare the trial to be futile, which means that there's a high probability that even if we continued to the end of the planned recruitment, we would not show the effect that the trial was powered for. And therefore, we decide it's not worth continuing. So an example here, if we analyse this trial 4 times, this particular one, each time that Z-score is getting higher, and after the 4th analysis there, we cross the top line, and we would stop the trial, saying that this treatment is beneficial. Now, the actual stopping boundaries are slightly more complicated than this, because that triangular shape actually comes from if you analysed after every individual patient. So for the real stopping boundaries, you get what's called a Christmas tree, and I'll show you that, and you'll see quickly why it's called it if we look at the actual stopping boundaries of the Eolia trial. So you have the triangle on the outside here, but the real stopping boundaries are these green dashed lines, the Christmas tree, which is because of waiting 60 patients for each analysis, you get that slightly greater power and slightly narrower stopping boundaries each time you analyse. So if we look then at those sequential analyses in Eolia, the first 3 times they analysed after each group of 60 patients, the result was inside that Christmas tree, and the conclusion is continue with the trial. But at the 4th analysis, that X, although it falls still inside the triangle, it's outside the Christmas tree. And this means that the recommendation at their 4th analysis was to stop the trial for futility. We're crossing the lower boundary, but with a quite substantially positive Z-score. The final point on the plot is the final analysis of the trial. Inevitably, because of these sequential analyses, they're continuing to recruit patients, continuing to follow up patients once the analysis is done. So even when you decide you're going to stop the trial, there'll always be a few more patients after that, which can then slightly change conclusions, although in this case, that final analysis also still fell in that futility area of the plot. So clearly what we expect to see then, we've stopped for futility, there's going to be no difference. They'll be exactly the same, won't they? But no. We look at the results now, and we see a 10% absolute reduction in 60-day mortality, with a 95% confidence interval from a 2% increase in mortality to a 22% reduction in mortality. So I think, and a p-value pretty close to 0.05, but we've got 0.07 here. And I think what we'll tend to think there is, well, that's a pretty big effect. It's close to being statistically significant. How can this trial be futile? Well, maybe it's the crossovers. I think there's been a lot of focus on crossovers in this particular trial. Use of ECMO was allowed in the control group. It wasn't should we just completely ban ECMO versus should we use it on everyone. It was ECMO as a first line in this, within 7 days in these patients, or use it late as a rescue. And there were more than a quarter of patients in the control arm did receive ECMO. So it is a potential issue to look at. But they got it on average 4 days after randomization. So I think that's worth bearing in mind in terms of whether these are really crossovers. It does look like it was being used as intended as a late rescue therapy. In the analysis and pre-specified, they did look at a particular statistical technique, which I won't go into any detail of, to try and look at what the effect would have been if there was no crossover, what's called a rank-preserving structural failure time model. And if we compare the results of that model against the primary results, looking at hazard ratios, you see you do get a slightly stronger effect using the rank-preserving analysis. So it gets a hazard ratio of 0.5, essentially, compared with 0.7 on the ITT analysis. But it's worth bearing in mind that this is quite a strong assumption of this model, that it assumes the treatment effect is the same for patients that cross over as if they'd been allocated to the intervention group. And that really is a very strong assumption, because these patients are crossing over after an average of 4 days, where presumably their treatment is not going well. I think to assume that ECMO is going to be as effective delivered after 4 days as delivered within 2 hours is a very strong assumption. So the fact that it hasn't really made a particularly large difference to the results, even applying that very strong assumption that the crossovers would do as well as if they'd been assigned to ECMO in the first place, means that I would say the crossovers didn't really have a big impact on the results of the Aeolia trial. Well then, so clearly it must be this fancy triangular design. That's the problem. So Aeolia stopped early. If only we'd carried on and they'd recruited the whole number, we must have got the result we wanted. But did it really stop early? So the issue with the triangular design is you have a trade-off. The reason why you want to use it is to get to a definitive result more quickly. And the trade-off there is there's a high chance that a triangular design trial will be shorter than a conventional trial, will require fewer patients. But the trade-off is there's a small chance, actually, that the triangular design could require more patients than a conventional trial. Now, in this case, Aeolia recruited 249. They had calculated that with the design, the maximum, if they'd gone to the end of that triangle, would have been 331 patients. So in that sense, it did stop early. But if they'd designed this as a conventional two-arm parallel group trial, using the same assumptions that they used in their sample size calculation, they would have recruited 194 patients, stopped the trial, analysed, and that would have been the data that we were looking at here. So although Aeolia stopped early compared to the maximum for the triangular design, actually it recruited more patients than a conventional parallel group trial would have done. So in fact, it was the tantalisingly close to significant result that was coming up on those first few analyses that actually kept Aeolia running for longer than a conventional trial would have run for. So unfortunately, the answer, as so often, and as we've already seen in some of the early ones, is power. The trial was designed with 80% power to detect a 20% absolute reduction in mortality. To frame that differently, it was designed with 29% power to detect a 10% absolute reduction in mortality. So if we think that 10% absolute reduction that we saw in their results is a potentially important effect, and I think most of us would think that in any group of patients, a 10% absolute reduction in mortality is an important effect. This trial was never powered to be able to detect an effect of that size. And as I said before, futility just means that it's unlikely that if the trial continued to its maximum sample size, it would show a significant result. And what that means is we were unlikely to get to a 20% absolute reduction in mortality, what was defined ahead of the trial as being that clinically important result that we needed to be able to demonstrate if people were going to use ECMO. We had to be able to show it reduced mortality by 20%. So I think I would argue that with these assumptions, Aeolia really was pretty much futile before it ever started. And as I say, I don't think it's the design that's at fault here. So that design works well if your trial is definitive. It's really important that the trial has to be powered to give a conclusive answer, even if if the answer is the treatment effect is not going to be 20%, that needs to answer the question once and for all. If the treatment effect will not be a 20% reduction, is not sufficient to answer your question, don't use a design that's going to stop futility. You would want to power for a meaningful effect over a full trial. So in that case, how do we interpret the result? Well, I think the first thing to look at, obviously, is what do the authors conclude? So among patients with very severe ARDS, 60-day mortality was not significantly lower with ECMO than with the strategy of conventional mechanical ventilation that included ECMO as rescue therapy. I find that a bit of a disappointing conclusion, really, on such a large and detailed set of analyses, that the whole thing is just obsessing around, is the p-value less than 0.05 or not? All we care about in our conclusion, it was not significantly lower, even though we've got a 10% effect size. And I'm not the only statistician that thinks that way. A couple of months ago, there was a really great commentary in Nature, titled slightly misleadingly, Retire Statistical Significance. And this was signed by more than 800 researchers arguing that the way we use statistical significance in scientific publications across many fields, not just medicine, is not fit for purpose. So to take some examples of what they would raise as why you shouldn't focus on statistical significance is obviously it relates to power again. So if you have two different studies here and you focus just on the significance, we say that this study showed a massively important difference and this one here has no difference. But actually we could be looking here at the same intervention, we have exactly the same outcome, the effect size is identical, but the conclusion would be that one is a significant result, one is not significant, and that's a difference and no difference. How can one be a difference, the other be no difference, when the effect is exactly the same? And you may think, well, no, that doesn't. People don't really report things that way. But within the paper, they pulled on a few different publications that have looked at this particular issue and found that looking at almost 800 articles, that's pretty much 50-50 as to whether the results were wrongly interpreted or appropriately interpreted in terms of focusing solely on significance and taking no notice of effect size. So if we followed their guidelines on how they would suggest the results of Veolia were reported, the conclusion may have looked more like this. So among patients with very severe ARDS, providing ECMO within 2 hours was associated with a 10% absolute reduction in 60-day mortality compared with the strategy of conventional mechanical ventilation that included ECMO as rescue therapy. So important to say what the actual best estimate of the effect is, even if that's not significant. However, you would then go on to couch that in terms of the compatibility of the data with potential effect sizes. And they suggest the terminology for a 95% confidence interval being that the results were reasonably compatible with anywhere between a 2% increase and a 22% reduction in mortality. So that would be a kind of a suggested interpretation of the Veolia trial on its own. But is there another way we could approach this? And I'm sure many of you know there is, because only a few months later, very quick off the mark, a group, including some of the key authors from the original trial, presented a post hoc Bayesian reanalysis of the Veolia trial. And this gives us a different framework that we can use to look at those results and try and make some more sense of them. So why would we want to be a Bayesian? The benefits really of a Bayesian analysis are that it takes account of either prior information or prior beliefs before the trial started. You then combine that prior information or beliefs with the data from the trial, and it provides you with interpretable results. And the assumptions that you make are all explicit. You have to lay out what are your beliefs before you do the analysis, and then you can see how the data affect those. So we take the prior plus the data, and we get some answers. So what is a prior then? Essentially, the prior just takes your uncertainty about the question that you're trying to answer with this trial and expresses it in terms of a probability distribution. So here is one of the priors that they used within that reanalysis, and this was their strongly enthusiastic prior. And what we have here is a probability density. So that shows us that their most likely estimate of the relative risk before the trial started, if you were this strong enthusiast, would be that a third of deaths would be prevented by early ECMO. So the peak of that probability distribution is at a relative risk of 0.67. And then this enthusiast is pretty confident that the real relative risk would be somewhere between 0.41 and 1.09. So 95% of the area of this probability density falls in that region. But they do have, they're not completely convinced. There's an amount of information over at this right-hand edge where they do think it's possible that the treatment doesn't work. If you went into this with complete certainty, if your prior is, I am absolutely certain that ECMO reduces mortality by a third, then no amount of data will change that prior belief. So we always need to be allowing for some uncertainty. And a good way to kind of quantify the uncertainty in these is to think about the amount of evidence it would be equivalent to. So our strong enthusiast here, their belief about the effect of ECMO is equivalent to if there had been a previous trial with 100 participants that showed a 33% relative risk reduction. And that's kind of how strongly they believe in it. So you could say that's not maybe that strong. You'd only need to have seen 100 patients to believe it worked. But this was the strong enthusiast. So then what we do, this is taking the figure now from the paper, we have in the yellow line, or the orangey line here, that prior that I just showed you. The shaded area here is the likelihood of the data. So looking at just from the trial alone, what values of relative risk would be most compatible with the data. And when you combine those together, what you get is the blue line, which is this posterior probability density. So if these were your prior beliefs, and this is the data, then the rational conclusion would be that that is how much it changes your belief. So in this case, the data has moved this quite a lot, because our belief was only equivalent to about 100 patients worth of data. We've now got another 249 patients worth of data. So the data are dominating. The posterior is a lot closer to the result from the data than it is to the prior, but it's being shifted down a bit from what the data say by our prior beliefs that it's strongly effective. So that posterior distribution allows us to answer explicit questions about the results of the trial in terms of probabilities. So here I've pulled out that posterior distribution from the previous plot. So this is our strongly enthusiastic posterior distribution. And what types of things can we get out of that plot? Well, the first thing to look at is obviously where's the maximum. So we can say from this plot that if those were your beliefs beforehand, and now you've seen the Eolia data, then you would now believe that the most likely relative risk associated with ECMO is 0.74. This is our posterior median. We can also look at the areas under this curve. So if we look at the total area below a value of 1, that essentially is our probability that ECMO reduces mortality among these patients. And if you came in with the strongly enthusiastic priors, then having seen the Eolia data on top of that, you would now believe there's a 99% probability that ECMO does reduce mortality by some amount. But actually we can go further. We don't just have to look at, is it less than 1? We can look at any area under this that we like. So, for example, well, my prior belief was 0.67 was the most likely. So how about now, what's the probability that the effect of ECMO is bigger than that the relative risk is less than 0.67? We just need to look at the area to the left of that on the plot. And we can say that this strong enthusiast now still thinks there's about a 22% chance that ECMO may produce an effect bigger than 0.67. 0.67, of course, is actually the effect that Eolia was designed to detect. So I think the investigators, by using this terminology, have somewhat outed themselves as being strong enthusiasts before Eolia started. And another thing that would commonly be used is that we can look at the middle portion of the distribution that has an area of 95%. So that tells us that there's a 95% probability that the relative risk is between 0.57 and 0.94. This is what we call in Bayesian analyses a credible interval. So some of those summaries have been included on this plot. So at the bottom here, we see that point estimate of the posterior median value, and the line shows us that 95% credible interval, so where most likely that the effect would lie. And it's an interesting exercise to compare this with what we get out of a standard frequentist analysis of a trial. So the p-value. All of these papers seem to love their p-values, but what really is a p-value? The p-value is the probability of obtaining the data, or more extreme, if there's really no difference in treatments. And that's a slightly strange and convoluted thing, really. That's not how our minds work. What we would really like to know is what's the probability that there's no difference in the treatments, given the data that we've observed, kind of reversing around the p-value there. And what you can see is that by using those posterior distributions, that is a question that we can answer from a Bayesian perspective. Similarly with the confidence interval, we almost always interpret confidence intervals in the way we would like them to be, as being an interval that has a 95% probability of including the true value. That's not really what they are. the 95% confidence interval says that if we repeated this study lots of times, this is where the frequentist terminology comes in, doing things frequently, then 95% of those confidence intervals would include the true value. It's not quite the same thing. And what we'd like it to be, as we say, is that interval with a 95% probability. We've already shown you that is a Bayesian 95% credible interval. So really, when we're interpreting confidence intervals, we almost always wrongly interpret them as if they were credible intervals. So what did they look at in this particular paper? Another advantage of that Bayesian approach is that you consider a number of different priors, and you can see how the data would affect all these different starting points. A Bayesian analysis will almost always include what's called a minimally informative prior. Essentially, I don't have any prior belief, any value of the relative risk is equally likely, and then really it will let the data talk, and you'll get a result that's very similar to the frequentist trial result. And then they looked at 4 different priors. A moderately enthusiastic prior, so that would have been equivalent to a previous trial that had 264 patients and a relative risk of 0.78. They chose that relative risk of 0.78 because that was the effect size in the ARDS-net low tidal volume trial. So that's saying that our moderate enthusiast believes that ECMO would have a similar effect to low tidal volume ventilation. And although they're the moderate enthusiast, actually their belief in that value is slightly stronger. That's equivalent to quite a large trial, in fact, larger than any modern day trial of ECMO. So it's quite a strong belief in quite a big difference, whereas the strong enthusiast has a weaker belief in a bigger effect size. So it's those 2 priors are a little bit hard to directly compare because, as well as differences in their expectation of how effective the treatment is, they also differ in the amount of information that they're bringing into the model here. And then they look at 2 different skeptical priors. They're not looking at any priors that consider harm as the most likely. That's a possibility. You could say if my prior belief is that ECMO is harmful, how would the results change it? But their skeptical priors are saying that essentially we have people here that believe there is no difference there. Their starting belief is the relative risk is 1, and then looking at 2 different amounts of information, one of which is equivalent to having seen a previous trial with 100 patients with a relative risk of 1, and another that's equivalent to a previous trial of 264 patients with a relative risk of 1. So all of these distributions are essentially plucked out of thin air, although there are some guidelines as to how to produce those, and that they can, taking relative risks that come from different interventions in similar patient groups, or ones that were used to design the trial. We already noted the strong enthusiast is looking at the effect size the trial was designed on, and looking at an amount of evidence that's kind of equivalent to a big pre-existing trial in this area, or a pretty small trial in terms of whether something's a strong or a moderate belief. And if we look at those priors in terms of their distributions, what you see there is that minimally informative prior is this blue line down the bottom, essentially saying all values of the relative risk are equally likely when I go into this trial. I have no prior belief. And then we've got the 2 different enthusiasts over on the left-hand side, and the 2 different skeptics here who are centered on the value of 1. And they also looked at some data-derived priors. So as I say, priors can be about beliefs, but they can also be about existing data. And where you have good existing data, people would often argue to focus on the data-derived. I think it's slightly questionable how good you would say the pre-existing data on ECMO were. And these data-derived priors came from a combination of 3 previous studies, only one of which was a randomized trial, the CESAR trial, and 2 observational analyses. I think both were in H1N1 flu, so maybe not so generalizable to the typical ARDS patient. And if you combine those 3 studies, what you get is the orange line here, which is actually quite similar to the strongly enthusiastic prior, and may argue where the investigators got their enthusiasm from. The relative risk is pretty close to 0.67. if you don't really believe that those studies are directly relevant to the question you're answering, then one thing you can do in this Bayesian framework is to downweight that evidence and say, well, yes, that's the evidence, but I don't completely believe it, so I'm just going to spread it out a bit. And I keep the point in the same place that's still coming from the data, but if you downweight the studies by 50%, you get this dark blue line, which is a bit wider, or if you downweight them to only 10% of the information, then you get that much wider estimate. And that just allows us to take less of the information through from the prior studies. And then what we get is that same plot for all of the different possible prior distributions they put on it. If you look at the minimally informative prior over here, what you can see is really the data are doing the talking. The yellow prior is flat, the shaded likelihood of the data, and the posterior, therefore, almost perfectly match up. The posterior distribution is just what do the data show. If we look at the three ones using prior information, those are these three blocks here. In all of these cases, these priors are quite strongly in favour of ECMO, and every time the data is pulling them back up towards one, but they all end with still quite substantial effects of ECMO. And actually a smaller amount of information or smaller amount of the distribution above one, so it is strengthening our belief that it has an effect, but reducing the size of that effect from our prior belief. And then we've got the four different reference priors at the bottom. So we saw our strong enthusiast before. The moderate enthusiast, actually, their prior turns out to have sat on almost exactly what the data showed. So the result is just they get a lot more confidence in what they believed in the first place. The point stayed in pretty much the same place, but the distributions got narrower. Whereas our skeptics started off believing there was no difference, but the data is having quite a strong effect, and it does pull that posterior distribution down below 1 for our two skeptics. And we can also do the same thing looking at an absolute risk reduction rather than a relative risk. So exactly the same format of plots. I won't take you through them. The conclusions are broadly the same. And then using those distributions, we can pull off some of those probabilities that we looked at before. So if we take the minimally informative prior at the top here, the results are pretty similar to what the primary analysis of the trial using a frequentist approach showed. So posterior median absolute risk reduction of a bit over 10, and that credible interval is pretty similar to the confidence interval that we saw in the original publication. But we can start putting some of these other posterior probabilities on it. And they look here at the probability that the true absolute risk reduction is greater than a number of different possible thresholds. And from that minimally informative prior, you would say there's a 92% chance that the posterior probability is that the true risk reduction is at least 2%. But it's very unlikely to be at least 20%. And really, this ties into why the Aeolia trial stopped. It got to a point where it was very unlikely that the true effect of the intervention was 20%. And that's what that triangular design was set up to detect. So these results are completely consistent with what that primary analysis is showing. But we can now see the effect of taking these different prior beliefs. And even if you started off with that strongly skeptical belief, you in your head have the equivalent of a 264 patient trial showing no difference, even though no such trial has ever been done, then the Aeolia results should lead you to think that, well, the effect of ECMO is likely to be at least 4%. There's a 63% chance it's at least 4%. There's a bit of a chance it's at least 10%, the 10% result we saw, although you'd be absolutely sure it couldn't be 20%. And we can answer those different probabilities. As I say, the ones using prior information look very similar to our strong enthusiast, because that's pretty much what the previous data showed. So what would the interpretation on these be? So we would say that across a range of prior beliefs, from strongly skeptical to strongly enthusiastic, the posterior median of the absolute risk reduction ranged from 5.6% to 12%. Even with a strongly skeptical prior, there's a 63% probability that the absolute risk reduction from ECMO is at least 4%. And even with a strongly enthusiastic prior, there's only a 2% probability that the absolute risk reduction is at least the 20% that Aeolia was designed to detect. And those data-derived priors gave even stronger results in favour of ECMO, but do rely largely on the observational studies, which may be biased. So in conclusion, while not reaching conventional statistical significance, the results of Veolia indicate that ECMO is very likely to be an effective intervention for very severe ARDS. And there is unlikely, therefore, to be sufficient equipoise for another trial in this patient group. And therefore, those are likely to be all the data that we ever get to inform these treatment decisions. Thank you very much.
Any questions? Yes.
Thank you very much for this brilliant presentation, very sophisticated. I have general comments, but open for discussion. To me, this study teach me that if we want to do this kind of study, we should not do only national studies. Because if the French group would have created a multinational, worldwide team for such an important study, it was very likely that they could reach at least the 600 patients that they planned. So this, I think, is a very important message, that when we plan such expensive and big trials, and we want to have some answer, we should have much more collaboration. Otherwise, I think that the future will not be anymore for a single-center trial that have no major impact on clinical strategies. Even national studies is almost impossible. I discussed with the French friends for other purposes. They cannot afford in the future more than this number of patients, whether perioperative or AVS. To me, the future is only worldwide big collaboration that can have some. I would like to have your opinion on that. It's time to have to move to another level of collaboration in this kind of randomized trials.
I completely agree with that, and I think a lot of work is moving in that direction, either through directly multinational trials or through harmonising a number of different trials across different settings, which can sometimes be easier from the funding point of view, because getting the funders to actually talk to each other and agree between different national funders to fund an international trial can obviously be a lot of hard work. But I think the single-centre trials are really only useful for hypothesis generating, and we have to move away from implausible results from a single centre going directly through into international guidelines telling everyone they should implement that intervention straight away, which then, as soon as that happens, it puts up barriers for the big trial that needs to be done, because that big trial is having to go against guidelines rather than what it should be doing, trying to confirm an experimental result. I think for interventions that are widely applicable to a large group of patients, there still is potential for national studies to be able to recruit enough patients. But as Luigi said earlier, we do need to be focusing our trials more down on meaningful kind of phenotypes and patients where a particular mechanism of action may actually produce an effect rather than expecting to get the same effect across all patients. And that is going to reduce the numbers of patients eligible in any particular country or setting, and make these multinational collaborations much more important.
Good morning. Thank you very much. Thanks for complex statistical slides and teaching. My concern with this trial is the timing. It doesn't really reflect our practice. So we have seen quite a lot of patients with severe ARDS by criteria, but if we ventilate with a conventional treatment, 12, 24 hours, they all got better, or most of them got better. We probably end up referring to ECMO center probably once a month or maybe more. So with this absolute risk reduction of 10%, I would question how many of those would have, not require ECMO, would got better with a conventional treatment.
Yeah, I think that's, as with any trial, it's necessary to select one intervention and one control, and therefore a trial will answer one question and can never answer every possible question. So I think that what that would mean is we don't have a trial that tells us whether the approach that we use to ECMO in ARDS in the UK is the best approach. We have here a trial that compared a much earlier, more rapid use of ECMO in one group to what was, well, I suppose you might argue that the control group in this is slightly more similar to how we use ECMO in the UK. I don't know, would you say that, is it a rescue? Not quite, but we're probably more towards the rescue than towards the within 2 hours. So maybe it should cause us to reconsider, are we considering ECMO early enough in these patients? And if we're not, do we need a larger provision of ECMO within the UK if we believe that ECMO within 2 hours does actually reduce mortality in these patients?
Well, thank you very much, David. I think that was really masterclass on Bayesian analysis, and I sort of understood it more today than ever. But I was just thinking about, partially to your point, in a way the randomization took care of that concept, and we know that almost 1 in 3 did require rescue ECMO, and it's very difficult to know exactly how to deal with this crossover. So 1 in 3 patients got late ECMO, and the mortality of this group was very high. I mean, I can't remember precisely, but it was like 55%, something like that. And the other thing to say was that they applied maximally protective strategies. So 90% of the patients were prone. Now, this is, if we look at the LungSafe trial, so international, 13%, I think from memory, are prone and I think in the UK probably will be similar or lower. So I think in order to compare this with clinical practice, we need to maximize conventional care. But I'm still concerned that 1 in 3 patients got crossed over to the other side as a rescue ECMO, mainly VA for cardiovascular dysfunction. So you talked a little, it comes to the question, talked about rank preserving. How do we see some of the trials who's analyze data intention to treat. So these patients would be as a conventional, and then they do reanalysis per protocol. But I'm never sure exactly what the pros and cons of that is. I'd be grateful for your...
Sure. So the intention to treat analysis is the analysis that will tell you the clinical effectiveness of the treatment strategy as delivered in the trial, or the difference between the 2 treatment strategies as delivered. And it's the only guaranteed, unbiased, provided the trial is well conducted, analysis. So I would always focus on the intention to treat, but that doesn't tell you, does ECMO work? It tells you, if we deliver this particular strategy of ECMO as delivered in the trial, and we can look at the timings and any patients that didn't get ECMO in the intervention group and whether they got it within the 2 hours and all those level of detail of adherence, but did that whole package give you a better outcome than the package of the conventional ventilation, including the rescue therapies, including the proning and all the other stuff. And when we compare those 2 strategies as delivered, what's the difference in the outcomes? Per protocol analyses, I would always advise to steer clear of, because they almost inevitably introduce bias, because the patients who don't comply with your protocol usually there's a reason for that. It's not, if it was just that the clinician didn't feel like doing the right thing and did that completely randomly, then a per protocol analysis could have some meaning, but that, I would say, is highly unlikely to be the case. Every clinician has a reason for going against a protocol, or for not delivering the intervention as it was designed, and anything that an individual does for a reason within a trial is going to introduce bias and break that randomization. There are approaches such as that structural preserving analysis, which in theory don't have that bias, but all of those require quite strong assumptions, and usually with anything involving crossovers, the assumption would be that the effect of the intervention among the patients who cross over would be the same as if they'd been allocated to it in the first place. Structural the rank-preserving structural failure time, that means that effect only gets applied from the point where they cross over onwards. But even so, if you believe that the timing of the intervention is important, then to assume that it's equally effective applied after 4 days as applied within 2 hours is an extremely strong assumption. But any analysis such as that requires some kind of assumption as to what would the effect have been in the patients who didn't get what they were allocated.
Any other questions? I have a question. Going back to what Professor Pelosi said, and I think you agreed that we should move towards multinational big studies and so on. What if we're asking the wrong question? What would this do also to innovation of ideas and sort of eliminate the groupthink that can occur? You know, historically in medicine, the groupthink has been wrong more than the innovators. So how do we deal with that?
If those big trials are designed correctly, then they can be used to weed out those examples. So as I say, I don't think EOU in any way is an argument against things like group sequential designs or other similar applications like adaptive designs and platform trials and so on of regular frequent analyses. If a trial is designed that it might recruit 2,000 patients and it's looking for a meaningful effect, so quite a small effect if it got to those 2,000 patients, then if it is this incorrect groupthink and a potentially harmful intervention, that trial won't get anywhere near 2,000 patients using a group sequential or an adaptive design. It will stop early saying, no, forget that intervention, it didn't work.
No, understood, but what if it's a question that is irrelevant? I mean, you may get a difference because we're not proving a causality. We're statistically analyzing 2 groups.
But with the randomization, ideally we are proving a causality. If we're applying it to a broad group of patients, we're not necessarily proving that effect size is equally applicable to every individual. And obviously designs that allow us to try and individualize treatment or at least vary treatment among subgroups would be beneficial in that respect. But hopefully the benefit of a randomized trial is it does show causality, because the only thing that should be different between the groups is that choice of intervention.
Well, thank you very much.